atomic level to molecular level. Then we went from molecular level to a cell level. And then from the cell level, we take it to a tissue level. And then from tissue to organ, an organ system, to the organism. So what we talked about on Thursday, we talked specifically about the atoms and then the molecules, of which we looked at the different types of bonds that held things together. We had the ionic bonds, and we had our covalent bonds. And we were able to differentiate between the difference between a polar and a non-polar covalent bond. Okay, do you guys remember that? And in our covalent bond, in our polar covalent bond, again, we'll kind of review a little bit because we're going to talk about a specific molecule that's going to be important in our cellular structure. And that is in our polar bonds, polar covalent bonds, we saw unequal sharing of electrons. And in our non-polar covalent bonds, what we would be able to see is an equal sharing of electrons. And those were our gases. We saw oxygen, we saw CO2, molecules that, for the most part, don't exhibit any electronegativity whatsoever. And the profound molecule that we talked about that had polar covalent bonds was water. And water exhibited hydrogen bonding, which was the third of our bonds that we talked about. And then we talked about our 4 macromolecules. Do any of you guys remember your 4 macromolecules?
Yes.
Okay, name them for me.
Carbohydrates.
Carbohydrates.
Lipids.
Lipids. Okay. Proteins.
Nucleic acids.
And nucleic acids. So they're the 4 main ones. So when we look at those, what we're going to be looking at today, we're going to be looking at these some of these molecules. And we're going to take those molecules, we're going to take a look at how they apply in cellular physiology. And going into, as a lead-in, let's take a look at our slides. Now what we're going to do is we're going to talk about our, once this thing loads up, we're going to talk specifically about our plasma membrane. We're going to be specifically zooming out of the molecules that we talked about. We're going to take it now to the cells, which are going to be containing those macromolecules. We're going to see those same things. We're going to see carbohydrates, we're going to see proteins, we're going to see lipids, and we're going to see DNA and RNA. And so now that we have a system of organization, we're going to be able to see how the cell is a basic unit of life, as you learned in biology. And we're going to take that, and we're going to look at the makeup of the cell, and we're going to understand the basic physiology of a cell. Ultimately, what we're getting at is that the cell is a factory on its own, and it's able to manufacture what it needs to manufacture. And for the most part, we're going to be talking about the production of proteins, and you'll be able to see that at the end of today. And as a background, when we look at today's lab that's due next Tuesday is going to be that fundamental concept of how we produce proteins. The same proteins that we talked about last Thursday, but you're going to learn how the DNA is tied specifically into them. So what we aim to talk about today, we're going to be talking about the protein carriers and channels embedded within the membranes. We'll talk about a concept of osmosis, which is the movement of water. Tonicity, which is all related to the concentration of a specific molecule in the cell. We'll take a look at the organelles inside the cell that help do what it's supposed to do. And then lastly, we'll talk about DNA replication. So by the end of today, you should have the concepts of at least of all the cellular stuff. That's the stuff that's going to be on quiz 1. So quiz on Thursday. That's my reminder for you guys. Quiz on Thursday, 6:45. You guys are going to have until about 7:20 to finish it. And then afterwards, we'll get into the lecture. Most of what you're going to be learning, and I will pinpoint the things that are not going to be of utmost importance, but you'll be able to see some of the other topics that we'll talk about as we go into our future lectures. So with that, we're going to go into our first cells. And so we learned in biology, we had prokaryotic cells, which were your simple unicellular organisms. And now our focus is going to be on eukaryotic cells. And these were more, for lack of better terms, more of your advanced cells. And these cells are going to be defined by having a true nucleus. There is a specific nucleus within that cell that's going to contain the DNA. And we have a nuclear envelope, and you'll be able to see this in our next slide over. Here you can see the nucleus. There's a center, just like we saw the nucleus of the atom, you'll be able to see the nucleus of the cell. This is the center point. And within that nucleus, you're going to be able to see the DNA that we're going to be talking about. And that DNA is going to contain the blueprints for that entire cell, that factory. And there's a very defined nuclear envelope, meaning there's a boundary to which the nucleus resides within. And also there's a cytoskeleton. And we'll talk about the nucleus, we'll talk about the cytoskeleton. But the cytoskeleton, much like the skeleton in our bodies, provides a support and shape of the actual cell itself. That allows it to have very defined shape as well as some extensions, which we'll talk about in just a little bit. So the cellular anatomy, as you can see, there's a nucleus, there's a cell membrane, which we'll talk about, the cytoplasm, which are all the contents within the cell, of which we'll see cytosol, which is this fluid, and the cytoskeleton, which is going to contain the filaments that are going to give it its structure. And then we'll talk about the organelles, and the organelles which will have a membrane and will not have a membrane, and we'll talk about those functions later on. Again, high-level overview of the cell. So the first concept I want to get into is the phospholipid, because this is the molecule that I talked about. And if you remember from Thursday, the phospholipid exhibits amphipathic or amphiphilic properties, meaning it loves both water and it hates water. And that is all inherent in the structure of the molecule itself. Here you can see that phospholipid. Remember, this was one of the lipids of which we had a glycerol backbone. Again, you don't need to know the structure, but what I'd like you guys to be familiar with is where the nonpolar and polar heads are. The fatty acid chains. These were long fatty acids with a carboxylic acid that's attached to the glycerol molecule. We had 3 attachment points, 1, 2, 3. 2 here are going to be attached to the fatty acid chains. And if you remember on Thursday, we mentioned that the fatty acid chains exhibit covalent bonds. But do you remember, were these nonpolar or polar covalent bonds? Remember? Nonpolar. Why are they nonpolar? Well, if you take a look at the carbon and you look at the hydrogens around, that electron sharing that happens very equally around. Remember, there's 2 elements that we talked about that are oxygen hungry, or electron hungry. They have a tendency to kind of pull the electrons and hold it much longer. Do you guys remember which ones those were?
Oxygen.
Oxygen and? Phosphorus. Thank you. So those 2 elements have a tendency to pull those electrons. Here we only really see carbons and hydrogens. Hydrocarbons are nonpolar covalent bonds, meaning those electrons are shared equally. And therefore, if we were to take a look at water, water doesn't have any point to be attracted to because there's no electronegativity. It exhibits no electronegativity. Now, on the other end, we would see our polar head. And attached to that 3rd spot on that glycerol molecule is that phosphate group. And here you can see the phosphate ion surrounded by oxygen molecules. Remember those 2 things we talked about, oxygen and phosphorus, are very electron hungry. And so this area exhibits a lot of electronegativity. And therefore water molecules are very attracted to it. So if you remember the structure of the water molecules, the electropositive portions, the hydrogen attached to the water molecules, will be attracted to the phosphate ions, or rather the polar heads. So they'll want to gravitate towards the polar head group. So this area would be polar, and this area would be nonpolar, hence the term amphipathic and amphiphilic. They basically mean the same thing. I mentioned earlier amphi, like amphibian, and philic, which is the love of. So the love of both water and non-water. So when we look at the structure of those phospholipids, and you dump them into an aqueous environment, I mean specifically a water environment, what's going to happen is you're going to see these phospholipids. And here we see the structure. And in the book, as well as the diagram below, you would see the polar head would be represented by the large head to it, and then the two fatty acids on it. And you'll see that arrangement take place, where if you dump a bunch of those phospholipids will start to arrange into this formation, where you get this phospholipid layer, and this is called the double phospholipid layer, where you get one layer on the outside, one layer on the inside. Why? Because we have an aqueous water environment on the outside and a water environment on the inside. So therefore, the polar heads would be exposed to the aqueous environments, and the nonpolar tails gravitate towards the middle of that bilayer. And this allows for that separation of that internal fluid environment as well as the extracellular fluid environment, of which I'm going to call them ECF on the outside and ICF on the inside. So this serves as the foundation where we're going to be seeing differences between that outside environment versus the inside environment. And if we look at the makeup of cells, this allowed sort of the evolution of cellular biology, where cells can exist in a, I wouldn't say a dangerous environment, but more toxic environment that the Earth had at its earlier phases, as opposed to the inside environment. So you have this barrier that keeps it protected from the outside world. And if we look at the makeup of the fluid from the inside and the outside, you'll note that these are going to be different. Here you can see the intracellular fluid, here you can see the extracellular fluid. And if you're going to look at the two, you're going to note some fundamental differences. And these fundamental differences, you just have to understand and you have to accept. So we're going to draw a cell where we have the ICF and the ECF. There's things that you just have to accept. And this is going to serve the foundation for neurophysiology, so this is going to be extremely important. Taking a look at the inside, the interior or the intracellular fluid, we'll see that the inside happens to be higher in the concentration of potassium. This is my notation for it. Increased concentration of potassium. Low concentrations of sodium. We've got a lot of organelles, which we'll talk about. We've got a lot of amino acids and proteins. On the outside, we've got the reverse, meaning we've got low potassium, high sodium, and we've got a lot of glucose on the outside. But the real main things that I'd like you guys to be familiar with are these: the concentration of potassium, sodium on the outside versus the inside. This is going to be also a good, another contributor to what we're going to be talking about in our neurophysiology lecture. So these are going to serve as the fundamental differences between these two. And so you can start to see that this is sort of the beginning of homeostasis. We want to make sure that the inside concentration remains high in potassium, and the outside wants to stay high in sodium. And for a cell to exist in homeostasis, for that cell to exist in rest, it needs to make sure that these concentrations are different. And we're going to talk about how that's done a little bit further into our lecture. So now that we have the differences between the two, you can see our phospholipids separating. Let's take a look and zoom in right on that phospholipid bilayer. Here, once again, you can see the hydrophobic fatty acid tails. You can see the polar heads littered both on the intracellular side as well as the extracellular side. And here you can see this is the extracellular fluid. This is the intracellular fluid. So again, as you would expect, outside would be higher sodium concentration, higher potassium concentration on the inside. Now, when you take those phospholipids together, they don't form a rigid structure. In fact, those phospholipids just kind of move around. They swish around quite a bit. And just due to the nature of having that barrier on the inside, aqueous environment on the outsides, these phospholipids literally just swim along. And if you were to kind of take your hand through it, You would eventually kind of swish these things around, they'd move around, but eventually they'd still maintain that barrier, hence the fluid mosaic model that was described in its onset at the very beginning when they described the cell. Now, in combination with it, if you were to see the phospholipids make up about 75%. You don't need to know these numbers. But phospholipids, about 75% of the makeup of that cell membrane. The other components you're going to see, about 20% of that, is going to be the cholesterol. And the cholesterol is theorized to maintain separation. It creates even bigger barrier between these phospholipids. And here you can see cholesterol. Again, one of our lipids with the 4-ring structure. This was specifically a steroid with those 4 rings. And you can clearly see that, 1, 2, 3, 4. And it's embedded within those phospholipids, and it allows for that fluidity in the phospholipid bilayer. Now, the other 5% are glycolipids. And these, you can see, are present. Glycolipids right here are protein, or actually rather, in this case, these are phospholipids with sugar, chains of sugar molecules or glucose molecules embedded at the very top. And they kind of look like antennae because that's exactly what they do. They allow for cell signaling, meaning they give characteristics to the cell itself. What do you mean by characteristics? Well, think about these. You've heard of blood types, right? Do you guys know your blood types? A, B, or O? Well, these are cell signals that tell you exactly what red blood cells might be comprised of, whether you're a type A, type B. It's a marker to basically tell the human body that these are human cells. In fact, these are specifically my cells. And these glycolipids serve as signaling just to say, don't attack me, so that the immune system doesn't come in and say, oh, I don't recognize you. If the immune system doesn't recognize it, it'll come up and literally will chew it all up. But if you have those markers on your cell, then it would preserve those cells. Kind of the whole concept of transplant. Why, if you were to get transplanted an organ and they want to match compatibility, what are they looking for? They're looking for cell markers. They want to make sure that those cells closely resemble yours in terms of the cell signaling, so that your immune system doesn't come in and attack it. Of course, they can't get 100% match. They might get 90, 80% match. But that's why people that have transplants, does anybody here know anybody that's had a transplant? Maybe one person? Transplant, people that have had a transplant have to take a lifetime dosage, not a lifetime dosage, but lifetime supply of immunosuppressants, meaning it suppresses their immune system from attacking that organ. Have you ever heard of organ failure or organ rejection? That's exactly what happens. These lipids aren't present on those cells, and therefore the immune system comes in and literally just attacks it. Autoimmune disorders work the same way. Those cells that might be produced might come in, and they might not see those glycolipids, and they'll come in and again attack those cells. So this is our way of doing it. So that's what we've talked about. Again, we've talked about our phospholipids, we've talked about our cholesterol, and we've talked about our glycolipids. The next thing we're also going to look at are proteins. And you can see that some of the proteins span through the entire width of the phospholipid membrane. And then you can see some other proteins that are kind of just embedded on the inside or maybe on the outside of the cell. So we'll talk about these proteins, but you can pretty much determine, or maybe not determine, you can sort of estimate what that function of that protein is based on the location. So by looking at whether the protein spans across or is embedded on one side, you can pretty much know what it might be able to do. And we'll talk about it a little bit those in just a little bit. So the next thing we're going to do is we're going to look at this concept of permeability. And permeability is the movement of molecules through something. While we know that the phospholipid membrane can maintain a separation of molecules from one side, the extracellular fluid from the intracellular fluid, we still need the movement of molecules and the movement of stuff inside and out. And how do we do that? Well, we have to look at the permeability of the membrane. And the permeability can change over time, and I'll talk about how that can get changed. Let's first talk about specifically how things move through. Well, the characteristic of the phospholipid membrane is that it is selectively permeable, meaning it only allows stuff to move through if it has things that allow it to move through. And the first thing we need to look at is the makeup of that molecule, hence why we spent time in understanding our nonpolar versus polar bonds. So looking at our permeability, the first molecules that can move through unhindered would be nonpolar uncharged molecules. So things like gases, we look at oxygen and carbon dioxide. O2, O, double bond O, and then CO2. C, double bond O, double bond O. These don't exhibit any electronegativity. Why? Because the molecules are shared equally. You might be thinking, well, Marlon, you mentioned oxygen. Oxygen happens to be electron hungry, but you get an oxygen molecule on one side, an oxygen molecule on the other side, and therefore there's going to be equal sharing. They're both electron hungry, but these are double covalent bonds which are nonpolar in general. Same goes with CO2. So with that, these gases move in and out fairly easily through the phospholipid membrane. They're small and they pass through. So these are our non-charged molecules. Now ions, on the other hand, things like sodium, things like potassium, they can't move through. Why? Because they're kind of attracted to the polar heads and they can't move past it. So therefore, ions don't cross through the phospholipid membrane. How do they move through? We'll talk about that in just a little bit. Other things, other large molecules, macromolecules, can't fit through. They're just too big. So even though the phospholipid molecules move through, that molecule just can't seem to squeeze right through those contact points. Other things to look at are lipids. Lipids have a tendency to be, lipids are actually nonpolar in nature, so they can slip right through. They're not attracted to the polar heads. They're somewhat attracted to the hydrophobic region, but they'll slip right through. So that is an important concept. Lipids, and then impermeable to ions, so things that are polar in nature. Polar molecules can't cross through. Non-polar molecules can. And so this will be important when we talk about in our endocrine lecture, how do we get things to move over and target the nucleus? So keep that in mind. Then the next thing we need to talk about is water. Water can move through. Even though water exhibits polar covalent bonds, and I did say that polar stuff can't move through, water is the exception. And if we look at water molecules have a tendency to be small. You would think, given the concept that we talked about where water would stick to the polar heads, it's small enough that it can actually fit through. So water is that exception, hence why water is one of those molecules that's widely studied, something that is of profound interest, not on just this Earth, but on our other planets. The last thing I want to talk about on this slide is the concentration gradient. So concentration gradient is exactly what I described earlier, meaning we have a higher concentration of sodium on the outside, we have a lower concentration of sodium on the inside. And whenever you have more of one thing on one side, that's going to give us a gradient. So if I can use an example, if I can imagine that my left side of the room is going to be intracellular fluid. Right? So you guys are the inside of the cell, and you guys are on the outside of the cell. And right through the center of this class is that phospholipid membrane. So with that, if I were to take all you guys, and all you guys are just random molecules, and we'll just say that you guys are, I don't know, sodium, for instance, and I cram you all onto one side of the room, in this case, on the extracellular fluid, there would be a tendency to want to diffuse across to the other side. So with this concentration gradient, that is going to be the driving force that will guide which molecules are going to move in which direction. So if I were to take, in this concept on our next slide, the diffusion capacity is tied into the concentration gradient. Let me redact that statement. That's one contributing factor. So diffusion capacity is the likelihood that things spread out. So if you remember that stink bomb example that I talked about on Thursday, I drop a stink bomb on the ground, right? You have all these molecules that have a sulfhydryl group attached to that molecule. It's going to break open, and now you get these molecules that start to diffuse across the room. When it's broken immediately, you might not smell it. But give it about 5 seconds later, all of a sudden you can start to smell it. Why is that? It's exactly tied into the concentration gradient. Within that stink bomb, you have a high concentration of those sulfhydryl molecules. The tendency of those molecules are to disperse and move across until they're equally spaced around the classroom. Something hot works the exact same way. This is the diffusion of heat. If I took a cup of boiling water and I placed it down on the table, eventually heat will start to disperse per the second law of thermodynamics, will disperse out into the room until it starts to heat up and eventually distribute equally. If I were to take all of you guys and cram you all into the corner, you'd all get uncomfortably close to one another. And if I say, okay, now you guys can move around, which way do you think you're going to move? Are all you guys going to move to the opposite corner and cram on the other side? No. You're all going to space out eventually until there's equal enough room. That's exactly what's happening with molecules in an aqueous environment. They have a tendency to kind of spread out. And you can see that on this top right image. You have a bunch of stuff dropped in there. And let me give you even another example. A sugar cube. A sugar cube is exactly this. You drop a sugar cube into water, and eventually it dissolves. They start to diffuse equally into that cup of water until all the molecules are all spread out. So when we talk about the movement, whether it's the movement of heat, whether it's the movement of molecules in an aqueous environment, we have to talk about the diffusion capacity. At what rate do these molecules disperse in an aqueous environment? So let's take a look at the contributing factors to those rates. So using that example again, intracellular fluid, extracellular fluid, right in the middle is our phospholipid membrane. If I have a bunch of you guys, and let's say you guys can move through that phospholipid membrane. If I had a bunch of you guys crammed into one side, let's say the extracellular fluid, the tendency would be exactly what I described here. You guys would want to disperse and move through the phospholipid membrane until you'd eventually equal out. Okay? Assuming that you guys can move through the phospholipid membrane. So the more people I cram to one side, the more likelihood that you're going to move in this direction into the intracellular fluid. If I have all you guys crammed on the intracellular fluid, which direction would you guys want to move? To the extracellular side. If you were both equal on both sides, do you get any movement? Actually, yes you do. The net movement is 0, meaning you get 3 you guys that move to one side, you get 3 that move on the other side. So net movement would be 0. So you guys are, I guess, technically right. There's no net movement. Now, what could I do to speed things up? What happens if I introduce more kinetic energy into this? Meaning, I add temperature in the form of heat. So if I start heating up molecules, what does heat do to a molecule? Kind of speeds it up, right? You're basically just a bunch of molecules that are just jiggling around in space. And if I start adding heat to it, those molecules move around faster. If I start making the solution even colder, eventually you start to slow down. You have reduced kinetic energy, and therefore less likely to move. And if you can, and kind of expanding on when we talked about our 3 laws of thermodynamics, the third one was absolute 0. And that's where I was talking about if you could get a solution down to absolute 0 temperature, that would basically mean the molecule stops moving. We can't get to that point just yet. So if I increase the temperature, I increase the diffusion capacity. Now, and steepness of gradient is going to be an increase. Now, mass, if I make the molecules bigger, right? If I make the molecules bigger, do you think that would speed up or slow down the rate of capacity? Slow down. This is simple physics. The bigger the object, the slower it'll move. The less likely it'll move across the phospholipid membrane. So this decreases. Now, the surface area of the membrane is the amount of space that you guys could diffuse across the membrane. So if I were to say your space to move across is only this big, and all you guys can only filter through that space, do you think that's going to speed up or slow down diffusion capacity? Slow it down. But if I were to increase the surface area, I'm going to make it the entire length of this room. That should speed it up, because you guys have more areas to move through. So the surface area of the membrane increases the diffusion capacity. Now, diffusion distance is the last one. This way, I'd be talking about not the surface area, but the width, how long it would take. So the phospholipid membrane is like this gelatinous substance. So if I were to take that width and make it even wider, would that slow down or speed up? Slow down. So the diffusion distance increases. So now you might be thinking, well, in what cases would you be able to see diffusion distance? Well, things like edema, and we'll talk about this a little bit more, but the buildup of fluid around the cell itself could start to thicken the width of that.
That would decrease it, right?
The diffusion distance?
Yeah.
So diffusion distance would decrease the diffusion capacity. If I were to make this thicker, that would mean that molecules would move slower. So you would be moving through that phospholipid membrane and then trying to move through this extra layer on the outside, eventually to make it to the inside or vice versa. So it's a further distance you'd have to travel through that barrier.
Would it be slowed down?
Oh, yeah, that's right. It would be right if I were upside down. That's nice. There you go. Good catch.
I tricky thing for them.
I want to trick you guys. So that, to recap, you would see the ones that would slow down would be the mass, the size of the molecule, and then the diffusion distance, the distance you have to travel. But the contributing factor here, especially in the physiological environment, is going to be this one, the steepness of the gradient. And you'll be able to see that when we look at our, which I call the YouTube example in just a little bit. So now that we've talked about molecules moving through, and we talked about, in the last slide over, we talked about selectively permeable membrane, meaning we've got this barrier and we have molecules that can either move through or not move through, where the most part, nonpolar molecules can move through. Polar molecules, such as charged molecules and ions, have a tendency to not be able to move through. Let's talk about how they can move through. Almost everything can move through with certain mechanisms. And we're going to take a look at two different types of diffusion. We have passive diffusion, and then a couple slides down, I have active diffusion, in this case, active transport. And the main difference between these two is it requires, well, at least active requires ATP. If something's passive, it just happens. If something is active, it requires energy, and that energy comes in the form of ATP. Remember, that's what we talked about at 9:35 on Thursday night. That was specifically ATP. It was adenosine triphosphate. That was that molecule that forms that energy currency. Now, passive diffusion, we have two types. We have either simple or facilitated. So one way I can draw this out, we'll talk about passive, and we'll talk about active. Passive can be divided into simple versus facilitated. Simple diffusion is exactly the mechanism that we were talking about, where things such as nonpolar molecules that have a tendency to be hydrophobic can just move across. So non-charged molecules will move across through the phospholipid membrane. It's simple diffusion. You move through the phospholipid membrane. The other is facilitated. What do I mean by facilitated? Well, think of what the term facilitate. Facilitate means something to help. Something to help that molecule move through. So here you can see these are our hydrophilic molecules. So things like our charged molecules and ions, for instance. They can only move through if they have something that helps it. And in those cases would be channels. Channels, think of any channel that allows boats to move through bodies of water. I failed geography. I didn't do so well in geography, so I can't really think of where there are channels in the world. But they allow boats to move through. Boats can't move through land unless there's a channel that allows them to move across. That's exactly what these things are. In this case, in the analogy of the boat, I'd be talking about charged ions. Now, carriers, on the other hand, are things that allow larger molecules to move across. And I'll have an example for that in the next slide over. But before I get to this slide, I do have to talk about what these helpers are. So what facilitated passive diffusion? These are those proteins. And let me go back to an earlier slide where I talked about the location of these proteins among the phospholipid bilayer. We have transmembrane, which span across the entire width of the phospholipid membrane, and we have peripheral ones where they just land on either the extracellular side or the intracellular side. And this I pointed out in our other slide. Here, this would be an example of a transmembrane or integral protein. The book uses integral protein. Transmembrane is the same thing. It's trans the entire membrane. And in these cases, transmembrane, if you look at the transmembrane protein, well, this is an image I've been dying to change forever because it's so blurry. But if you look at the protein, you can tell the function of that protein as a facilitator based on its location. So if the protein spans both the intracellular side and the extracellular side, it must mean it's doing something on both sides. And that's exactly what these ion channels do. They allow specific ions to cross through the membrane. And these ion channels are attuned to the specific ion. So we'll talk about sodium channels, we'll talk about potassium channels, and they only allow that ion to move through. Now, carrier. These are for the other facilitated diffusion. Carrier are transport proteins that allow molecules to move across, but they literally have to carry it from one side to the other. And so the term that you most likely, I know most likely you're going to be able to see this, ligand. The term ligand is a molecule that binds to a carrier. So be familiar with that term because we'll be throwing this term quite a bit, ligand. A molecule that binds to the carrier. And let me give you the example of a sugar molecule on the next slide. In order to get glucose into the cell, it's a large molecule. It tends to be polar, so it can't move across the phospholipid membrane on its own. So this transporter allows glucose. Remember, glucose is high on the outside, and we need to get it into the cell in order to generate ATP. If you're wondering how, we'll talk about that on Thursday. We need to get glucose into the cell. We've got a glucose transporter that binds glucose onto the active site on the extracellular side. Once it binds through, we're going to get this configurational change that allows glucose to spit it out to the intracellular side. So here, because we can see the gradient exists from the high concentration of glucose on the outside to the low concentration of glucose on the inside, these carriers allow that movement through one side to the other. So this is one of the things that you're going to be able to see. So you might be wondering, well, does that mean we're going to take all the glucose on the outside of the cell and shuttle it inside? Well, there's a way to modulate this activity. And to kind of give you an example, we kind of talked about it on Thursday. There's a way to stop this glucose transporter. We don't want to take all the glucose that's available outside and shuttle it in. We only need to take what we need in. And the way we can do that is we use something. So if this is our active site, and I know I'm using these terms kind of loosely because this isn't necessarily an enzyme, there are other sites, kind of like allosteric sites, that can come in and activate this transporter. And if you're wondering what that thing that would bind to this thing right here, well, one of the things that is a common thing to activate these glucose transporters is insulin. So if I were to use that example earlier, this is a phospholipid membrane, and I'm trying to carry all you guys. You guys are glucose molecules. I'm wanting to move you guys one by one. Something needs to tell me to stop. No, we've got too much glucose on the inside. So if insulin makes its way and attaches to me, that's when I turn on, and I can now move molecules in. And eventually, when that insulin is taken off, then I stop. Okay? So insulin is a common thing, and the direction of which of glucose moves, and nothing you really need to know for now, is really dependent on which cell we're talking about. Right now, we're just talking about the generic cell. But you'll be able to see that there's different types of glucose transporters. And this example that I'm using, again, not something you need to know, but this is GLUT4. This is one of the glucose transporters that is activated by insulin that allows glucose to move into the cell to be used. And these are common in muscle cells. You would see these typically where if a muscle is active, it needs sugar in order to start generating energy for the cell. There are also glucose transporters in the liver, in the kidney, small intestine, for instance, another thing where we're going to have extracellular fluid and we want to get glucose into the bloodstream. So a different type of transporter. And what would be the main differences between the glucose transporters is just the makeup of it, the structure of it, the way it's folded. Again, something that we'll talk about a little bit later on today, and you'll get an idea of how we encode the blueprints, the primary sequence for this protein that will lend itself to the secondary sequence, that will lend itself to the tertiary sequence, and then putting all those together, our quaternary structure for putting that protein together. Again, tying what we talked about on Thursday. So if you were to think about ways to increase, and let me tie this into, for instance, exercise. Okay. If we look at the generic muscle cell, and we know that sugar molecules, or glucose molecules, are used to generate ATP on the inside of the cell. We would think that we would want to now be able to shuttle glucose into the cell a little bit faster. If you're doing things like resistance training, if you're doing activities that require a substrate in order to generate ATP, we need to get energy, or we need to get a molecule, glucose, faster into the cell. So looking at these mechanisms, what ways can we increase the amount of glucose moving in? Well, one of the ways, we can increase the number of carriers. So increase the number of carriers. The more carriers we have, the faster we can shuttle glucose in. Can you guys think of another example? It might take a second, but kind of chew through it.
Increase insulin.
You can increase insulin. That's another response. So increase insulin, your target molecule. We can reduce the thickness of the cell membrane, even though I can't really think of a relevant example of where that would take place. Can I increase the temperature of the muscle cell? How do we do that? When you're exercising, are you cold all the time? No, you generate heat. You generate heat, we increase the kinetic energy. That allows glucose to move faster. We get those carriers to move faster. We're not doing anything to the mass, that glucose is going to remain the same. And then the other, the steepness of the gradient. We get glucose more readily available on the outside. So therefore, if we're increasing diffusion capacity through these channels, we get more glucose into the cell. You get more glucose into the cell, you can generate energy efficiently. And again, we'll talk about the mechanism of that in 2 days. With that being said, what questions do we have? So So last thing I want to look at for our integral proteins are the peripheral proteins. We've talked about the transmembrane proteins that allow movement in and out of the cell. Now the peripheral proteins are things that are bound to either the intracellular side or the extracellular side of the phospholipid membrane. So we have either enzymes of which we'll talk about, that perform actions on that side of the cell. The other ones that we're going to talk about are receptors, where receptors can exist on the outside of the cell, and they receive a signal of some sort, and that signal can be a hormone. Again, we'll talk more about that in our endocrine lecture, things that allow hormones to bind to the cell itself. We've talked about passive diffusion. Let's talk about active transport. And the key thing that describes active transport is the term active, meaning it requires ATP. It uses energy in order to move ions against their concentration gradient. Why would we do that? Well, we're going to learn that there is sodium on the inside. Well, we already know that. There's sodium on the inside of the cell. How do we get, if we start to see an increase in the sodium on the inside, and again, we'll talk about that next week when we get into our neurophysiology lecture. If sodium starts to come on the inside, how do we get it back out? We have to pump it across, and we're moving against a concentration gradient. So if the concentration gradient for sodium is towards the inside, and potassium, concentration gradient is going on the outside, in this case, this would be the intracellular fluid, this would be the extracellular fluid. We are pumping out of the cell, so against the concentration gradient. And we're moving, sodium is, we're moving sodium against its concentration gradient, and we're moving potassium against its concentration into the cell. And that requires energy. If you were to look at levees, they basically work the same way. Levees where you have water, you have a high level of water, and you've got a low level of water, and you're trying to get water back onto the higher side. You've got to use electricity to pump that water against. If I wanted to go the other way, well, you can just open up channels and just allow water to move in. This way, you require energy to pump it across. And the best example of that is the sodium-potassium pump. Again, we'll talk about this next week. But that sodium-potassium pump has a third attachment point, which is the ATP ACE. And the ATP is used as the energy to pump them across the gradient. Whatever sodium's left over gets pumped across, and whatever potassium is on the outside gets pumped back in. So now we've taken a look at different types of channels. We've looked at different types of channels and carriers. The reason why there's a lot of, at least for a good chunk of studying pathophysiology, and when you guys do get into pathophysiology, you'll start to look at pumps, you'll start to look at the dysfunction of pumps and dysfunction of channels as a setup for why diseases take place. And that's a whole term that's been used, that's been thrown around quite a bit, called channelopathies. One of the things they want to do is study the dysfunction of that particular pump or channel as a major cause of a particular disease. To give you an example would be seizures. When they look at seizures, they note that there's a dysfunction of these potassium channels. If you look at cystic fibrosis, there are chlorine channels. And I've talked about sodium and potassium, but there is chlorine also. There's a bunch of other ions. Chlorine channels are dysfunctional with people with cystic fibrosis. And therefore they get a buildup of chlorine, and that disrupts the electrical balance. And what do I mean by electrical balance? That's something we'll visit next week. But to give you an idea behind all this is to look at what happens with these ions. So if I were to draw our bilayer, and I have, well, let's just look at sodium, for instance. Well, actually, let's look at potassium. Potassium is high on the inside. We have low potassium on the outside. So if I get a bunch of potassium ions, potassium has a positive charge. If I have more positive ions on the inside of the cell in respect to the outside of the cell, that makes this side more positive, makes this side more negative. And we'll explain this concept in much more detail next week. And that lends itself to a certain electrical difference, which we describe as a voltage between the two. Again, if you're not getting that now, that's okay, because we'll revisit this next week. So in preparation for our neurophysiology lecture, I'd like you guys to be familiar with this voltage gating that should take place, because I've seen people get stuck on this whole idea. And in these ion channels, specifically a sodium channel, it requires a certain electrical difference from both sides in order for it to operate. And we're going to talk about the changes in the voltage in that phospholipid membrane. These voltage-gated sodium ion channels require the differences in charges that allow it to open up. So looking at the makeup of the sodium ion channel, and I'm just using this example here so that you can understand the fundamental concepts of charges and what opens these things up. We learned about insulin opening up glucose carriers. Here, sodium ion channels are activated by voltages. So on top, you'll see activation gates. This one's another gate called the inactivation gate. And at rest, these channels are closed. And once we get to a certain voltage, meaning negative on the outside and positive on the inside, and eventually there's going to be a certain change where you're going to open up that activation gate. And as long as that inactivation gate is open, you're going to get ions to move through. And eventually, once you get the ions moving through, that's going to cause a change in the voltage that's going to result in this inactivation gate closing, which is going to prevent things from moving through. That might not make sense now. I can tell by some of your faces you're looking at it and you're like, what is he talking about? That's okay. We're going to revisit this when we talk neurophysiology lecture. We're going to be talking about a specific type of cell. Right now we're talking about generic cell. We'll be talking about a nerve cell that's going to exhibit these properties that's going to allow for the voltages to change. So stay tuned for that. So with that, let me see. Okay, let me finish this portion up, and we'll take a break before we get to this one. So we've talked about the movement of molecules through a cell. Now we have to talk about how things move in and out of the cell. How do we get things in and out that are much bigger? And here we have to employ what are called vesicles. Vesicles, just like the term, I guess, somewhat similar to vessels, are basically sacs made from the cell membrane. So yes, they contain phospholipids. And the way we would do that, and I'm going to show you a slide on the next slide over, showing you an example of how we get things into a cell. So what we're looking at here is a microbe, and this is a fairly large, obviously not something that should exist in the human body. This is a cell that needs to get broken down. And what cells do is that they can take in things from the outside world, things that move into the cell. And this is what we call endocytosis. If you look at the term endo means into. So things moving into the cell. The exact opposite is exocytosis. Things that move out of the cell. This example is an example of things into. So this is endocytosis. And endocytosis can get broken down into two terms, phagocytosis or penocytosis. And the fundamental difference between these two, phago meaning to eat, peno meaning to drink. So phago is to eat large particles, peno is to drink large amounts of extracellular fluid. So whether we're talking about particles or we're talking about fluid. And in this case, this is exactly what phagocytosis is. We get a microbe that comes in and attaches to the receptors somewhere on that membrane. So it comes in and attaches to. And then we get, and you don't have to know about these pseudopods, but these basically will wrap around. And they wrap, the whole cell wraps around that microbe. And, no, don't restart. We get the cell membrane that wraps around. And what it does is it contains that microbe on the inside, so it can't really do anything. That cell membrane wraps around, you get this phagosome that's wrapped, you get the microbe that's wrapped around by that phagosome, and then you have these lysosomes, which I'll talk about later on. These are digestive enzymes that will come in and fuse together with that phagosome to come in and chop that microbe into a bunch of pieces. This is our way of a cell saying, I don't recognize this thing that doesn't exist. Let's come in and swallow it whole, break it down, and now you've got all the components of that cell. It's a way for cells to kind of eat something out. Penocytosis, on the other hand, is just basically a bunch of fluid. There's a fluid that's like high in glucose, or a bunch of glucose molecules in that area. Penocytosis, I don't even use the word glucose. Let's say just say some ion that comes in. We want to take a good amount of it in. That can get wrapped up and get swallowed in. That's penocytosis. Exocytosis, we'll learn later on. These are ways we can get things like proteins to make its way out of the cell. So we'll use that example later. What questions do we have?
So basically, this is how they kill cells that are foreign to the body?
Exactly it. Exactly. It's much more complicated than this, but the whole...
Does that have to do with the lymphatic system?
Yeah. There's immune cells in the lymphatic system. If you're looking at specifically lymph nodes, there's ways to do that. But the immune system, and one of the things I really wanted to get into is immunology, where we talk about the immune system and the cells that are involved in eating things up, things that don't exist. This is essentially how we get rid of foreign bodies in our system. It's through this process of phagocytosis. We come in, chop it up, and we use those parts. Any other questions? Yes?
So, the ion that you're saying there that is a part of penocytosis, it sort of like drinks it in? Is that what you're saying?
That's all it is. It's just ways for cells to take in a good amount of fluid to bring it into. Where we left off, we talked about the movement of ions, the movement of molecules through the cell. We've talked about the mechanisms of passive diffusion, active transport. Now, what we do need to talk about is the concept of a metric that we need to talk about in terms of how much stuff is dissolved in an aqueous solution. So this is something that you would have seen in chemistry. We don't touch too much on this. Dr. Fink goes over this quite a bit. And he goes into it because this is all related to drug administration and stuff like that. He has a specialty in that, an interest in that. I don't get too much into it because this is stuff that you would have seen before. In fact, I don't even really talk about this term any more than what it is listed on this particular slide. But when you measure a solution, and a solution has two components. Number one, the solute, what is dissolved. The solvent is what is used to dissolve that solute. So to give you an example of sugar water, sugar is the solute, while the solvent is water. Hence why when we talk about water, aqueous environments, water is that universal solvent. And when we were looking at salts, we talked about sodium chloride, table salt. We have sodium and chloride ion that are bound together through an ionic bond. And when immersed in water, will get separated. You'll get the separation of these ions. So you're going to get sodium ions, you're going to get chloride ions that are going to be surrounded by water. because our chloride ion, which is an anion, a negative ion, is going to get surrounded by the electropositive hydrogen atoms of the water molecules. Conversely, we're going to see our sodium. Sodium is going to get split up. It's going to get dissolved, and that ionic bond is going to break because it's going to be surrounded by water molecules where the electronegative oxygen atoms of the water molecules will surround that sodium ion. And then we're going to get it dissolved. So the way we can measure how much stuff is dissolved is not to just look at the concentration of the ion, but the term osmolarity. And the osmolarity, when we look at osmolarity, is the number of grams of our solute per liter of solvent. And this is going to have relevance in our urinary lecture. Towards the end, actually one of our last lectures, we talk about osmolarity. This is how much stuff is dissolved in water. And what we're talking about is stuff that is making its way that's going to be formed in urine. We have to look at the osmolarity. So when you see that concept again much later, it's the amount of grams, the weight of that molecule in the liters of solvent. Osmolality, we don't even really talk about. Osmolality is something that you see in chemistry, and that's just taking weight divided by the weight of the solvent. But since we're only really talking about water, we're not looking at anything else. A liter, you don't need to know this, but 1 liter of water equals 1 kilogram, so they're interchangeable. So in osmolarity, when we're talking about water, is really the only clinically relevant term that we're looking at. So we've talked about solutes, we've talked about ions, we've talked about molecules. The other thing that we need to talk about is this, and this is an important concept. Osmosis, the movement of water. Now when we go back some of our earlier slides, we've talked about things that move through carriers, our transmembrane proteins. Macromolecules not being able to move through. We've talked about carriers and channels that allow molecules and ions and other smaller molecules to move across through facilitated diffusion. But what we do need to talk about is the movement of water, because this is how we get water to move in and out of a cell. So let's take a look at osmosis. And water moves through 2 main mechanisms. The main mechanism is water squeezing in between those phospholipids. Remember the fluid mosaic model? We have phospholipids that are kind of swishing around. And every time they swish around, it creates a little space that water can kind of sneak through. It actually can even penetrate through that hydrophobic barrier, the non-polar fatty acid chains right in the middle of that phospholipid membrane. So it can actually move through. And then the last piece right here is a good bulk of water can move in through specific water channels called aquaporins. And these aquaporins are proteins that are embedded that can be embedded in the phospholipid membrane to allow water to move in and out faster. So we have the ability to get water in and out through a phospholipid bilayer by employing these aquaporins. We can activate aquaporins. We can build aquaporins and we can put them, bless you, right onto the phospholipid membrane to allow movement of water, and we can adjust it accordingly. In what case do we need to do that? Again, we talk about that in the urinary lecture. If we need to get rid of excess water, say you just drank way too much water today, you need to get that water out of your system, well, those urinary cells that we'll be talking about have the ability to build aquaporins to allow water to move out. If we need to conserve water, let's say you're out in the desert somewhere and you only have so much water. You're probably not trying to piss any of it out. You need to turn those aquaporins off. So water can move through, but aquaporins, we have the ability to modulate how much water moves in and out. Now the thing about movement of water is it just doesn't do it on its own. It's really guided by the solute concentration, meaning how much stuff is present in the water that allows water to move. And what do I mean by that? Well, let's take a look at our next slide, because that's going to help explain that concept. And this is the concept that I call the osmosis, or my U-tube example. U because this is the letter U, not Y-O-U, but the U example. And in this example, what we have is a tube with open channels at the top. We have a left arm and a right arm. And we have water that's filled up to the same level. And there's a division right in the middle, which is called our selectively permeable membrane. It exists right in the center, and it separates the left arm from the right arm. Now, it's permeable to water, meaning water can move in and out, but it's impermeable to the solute. And the solute is drawn on here by these yellow diamonds. So the yellow diamonds, our solutes, cannot move through. So when we look at this, we want to understand which direction, what pressures are involved to get our water molecules to move from one side to another. So when we look at their starting conditions, like I said, equal on both sides, water levels equals on both sides, selectively permeable membrane that only allows water to move. The fundamental concept that is important for you to understand before we get into this is that water wants to always move in the direction to balance out the concentrations on both sides. Meaning, if I had a salty concentration on this side, and I had a non-salty concentration on this side, meaning all salt on this side, the direction of water would be moving in this direction to balance out the concentrations. And this concept we'll talk about on the next slide called tonicity, which I'll get into in just a bit. So the idea is that water wants to move to balance it out. So if I had a couple diamonds over here, I had, let's say, 10 diamonds to the right, 2 diamonds to the left, water wants to move in this direction to balance out those concentrations. And eventually, what you would see is that water would want to move to the right. And that movement of water through that selectively permeable membrane is called osmosis. Now, looking at this example right here, you would think that water would keep moving. It would keep moving so that those concentrations would be equal, where, and again, I drew those 2 diamonds earlier. If I gotta draw again 2 diamonds over here, 2 diamonds over here, eventually the concentration here, where we have the amount of solute, is equal with this amount of water versus the amount of water here with those 2 solutes. So those concentrations would eventually match, right? So let me, by erasing those diamonds, same concept. Water would keep moving, but not all the water would move to the right. And the reason being that water wouldn't move to the right because this is a pillar filled with water. Eventually, you're going to get all this water that you're constantly pushing, and eventually we're going to get this push back. Because this water column right here is going to exhibit a pressure back. And that is called hydrostatic pressure. So hydrostatic pressure is due to the large volume on the right side, and eventually those water molecules can't push back. And that pressure is called hydrostatic pressure. So if osmosis is the direction to the right, hydrostatic pressure is the pressure pushing back due to the weight of the column back to the left. Exactly. There's a lot of water in this. There's a higher pressure on this side due to the weight of that water column. So what happens is we're going to eventually reach an equilibrium where the net movement of water to the right is equal to the net movement of water to the left, meaning that the osmotic pressure, the tendency of water to move to the right, is countered by that of the hydrostatic pressure that's pushing to the left. So this is at equilibrium. Now, looking at this concept right here, the one on the right is what's employed to restore starting conditions. And this pressure is the osmotic pressure. This is the pressure that's exerted to push that volume back. It's a resistance to osmosis. And to think of an osmotic pressure, I want to use an example here that could be used, would be looking at a cell. Okay, so I'm going to take this concept and apply it back to the cell, and we'll revisit this later on to see if you are lost on anything. Now, we're going to take this example earlier where we look at a cell. This is our cell, and here is the water, and we're going to call that ECF, the inside being ICF. And this example here of the ICF, or the cell, is the right side of the tube. This would be the inside of the tube, the ICF, and this side would be the ECF. Eventually, when you get the movement of water into the cell, if you get water moving into the cell, it's like a balloon. It'll start filling up. And eventually you start filling up, and then once you fill up, you're going to get this pushback. That pushback is the osmotic pressure. That's the applied pressure back on the system to keep that cell the same size.
I know this off topic, but does that correlate to edema? How if the cell is imbalanced or something, then the cell would.
Kind of. That's really more tied to the diffusion distance, but it is tied to edema in that if
like if they're not balanced, then once I'm assuming that.
right. So what happens when a cell contains too much solute? Water has a tendency to kind of move into it and cause it to swell. That's where you get this swelling. So edema, exactly what we're saying, is that's exactly what's happening where you get the movement. So the movement of water is the osmotic pressure. Now that the cell is filling up, that's hydrostatic pressure. That's pressure countering osmotic pressure. And the cell, the phospholipid membrane, employs the osmotic pressure to prevent those changes taking place. Now, let's take a look at the term of tonicity. And tonicity is referred to, the easiest way to refer this, is the extracellular concentration in relation to the intracellular fluid. In other words, tonicity refers to the solvent, the aqueous solution on the outside. And it wants to measure how much solute is in the aqueous solution compared to the inside. So ideally, what we would want for a cell is the solution, or the concentration on the outside matches that of the inside. Taking back that YouTube example earlier, we want the concentrations of solutes. It doesn't have to be the same solute, but the concentrations of stuff on the inside equal to that on the outside. Therefore, the movement of water, osmosis, is balanced with hydrostatic pressure. In other words, net movement of water equals 0. That's an isotonic solution. Hypotonic and hypertonic represent the imbalance to that. So hypo meaning low, and hyper meaning high. Hypotonic means low extracellular solute concentration. So the outside would have a low concentration of solute compared to the inside. Hypertonic solution would be a high concentration of solute on the outside in relation to the inside. And these have a fundamental influence on the cell structure itself. So to give you an example is an example that I've seen before where you take cells. Before I get into that example, let me talk about what a hypotonic solution does. So if we have a low concentration of solute, the direction of water, we have, and the best example would be here, we would have, if this is the extracellular fluid and this is the intracellular fluid, the direction of water wants to go in. And in this case, that cell is going to want to keep filling with water up to the point that the cell starts getting real rigid. And it's overfilled with fluid. Just think of it as a balloon. It's going to fill up and essentially could burst. If you keep trying to fill up a water balloon and it takes it way too much, that cell can burst. On the other hand, here if we have a very high concentration of solute, water wants to move in the direction out of the cell. We're looking at it from this opposite perspective. So it would be more salty on the outside. Water wants to go out. So if this is a higher concentration of solute compared to the inside, water wants to move out. And in those cases, those cells shrink. And if they shrink, they undergo what's called crenation. It's a shrinkage. So the example that I've seen before of this is if I remember seeing the examples of people that would buy a fish and they know nothing about fish. And they'll take a saltwater fish and put it into fresh water. Right? So you'll see what happens eventually to that fish. Fish has its, that particular saltwater fish has solutes in the cells. And it needs fresh water. It needs an isotonic solution to survive, meaning the concentration of the solute on the outside matches that on the inside. So take a saltwater fish and put it into fresh water, and what happens is the solute on the outside is low. And therefore water goes into the fish's cells, and eventually it'll cause them to burst. Now take the exact opposite. Put yourself in a hypertonic solution. Take a goldfish and put it in salt water. And what happens? Water wants to leave the cell, and the cells start to shrink. So that's obviously not good either. We want a balance, and that balance is isotonicity. We want a balance on both sides. Another example, if you've ever been dehydrated and you go to the hospital, they just give you a bunch of water? No, because if they give you a bunch of water, well, eventually what happens is you get into a hypotonic solution. They've actually done this before. There's a radio station that I remember hearing the reports of. They used the example of, and I don't want to digress too much, they had, when the Wii console came out from Nintendo, it was called something they called Wii for a Wii. And they wanted people to drink like as many as I think like a gallon of water in the shortest amount of time to win the Nintendo Wii. And they had this lady that called up, and I think she had drank like 2 gallons of water in like 20 minutes. And on the air, you start to hear her start to mumble, and eventually she doesn't say anything, and they cut the audio feed from that point. What they found out was that lady died because she had taken way too much water. And you think of those brain cells, and those brain cells are immersed in an aqueous solution. What is that aqueous solution? It's blood. Eventually you'll bathe in this extracellular fluid. And you start filling it with more water. That's going to decrease the concentrations of the solutes in the extracellular fluid. And what happens with those cells is that water starts rushing into that cell. And eventually those brain cells burst. And she died. And yes, that radio station was held liable. There was a huge lawsuit behind it. And they pulled it. They were like, never do that again. Clearly not a good thing. To take that example that I mentioned earlier, if you're dehydrated, what do? They give you saline solution. The solution that they give you, they want to match the tonicity of the extracellular fluid in order to get you to replenish those fluids. So therefore, by giving you an IV fluid of saline, they can still maintain isotonicity and still replenish the amount of fluid into your body. If you think about also long-term exercise, you would have sweated out quite a bit, you would have lost a lot of water, and you would have lost a lot of solutes. You can drink water, but they give you, if you've lost a lot of fluid, they can give you something like Gatorade. If you look at Pedialyte, you look at any of the other solutions they give you, it's a saline solution. It's salt and sugar mixed into water to maintain tonicity so that you can still increase your fluid without disrupting the mechanisms of the cell and the cell structure by avoiding hypotonic or hypertonic solutions. Okay, what examples do we have about tonicity or this YouTube example earlier? Everybody good? So the next slides, I know we've got quite a bit of slides to go over. I'm going to kind of breeze through. These you would have again seen in biology. What I'm going to be talking about for the next several slides are just the structures of the cell. Now that we've talked about the movement of ions, the movement of molecules, movement of water, let's just give a background of everything else in the cell. So cytoplasm is basically everything underneath the cell membrane. It's the fluid-like jelly within the cell. Take apart that phospholipid membrane, and you have the cytoplasm. And within the cytoplasm, you're going to get cytosol, which is the intracellular fluid, or ICF, of which a majority of it is going to be water. 75 to 90% water. The other stuff are going to be the dissolved substances, namely our potassium. We've got a little bit of sodium in there. We've got a little bit of glucose. We've got a lot of amino acids. We've got fatty acids in there, some waste products. We've got a bunch of proteins in there. It's everything dissolved in that. The organelles are the other cellular organs that we're going to be looking at, and the cytoskeleton being the filaments that are involved to lend itself to the structure of the cell. Looking at the cytoskeleton, we're going to be able to see that this provides that internal framework, of which we have three main parts: microfilaments, intermediate filaments, and microtubules. Don't worry about these. I'm never going to ask you about the types of cytoskeletons. In a cellular physiology class, yes, you would need them. But to kind of explain what they are, microfilaments are basically thin filaments of actin. And what they do is they lend itself to the structure of the cell. So if you were to look at these villi, these are projections on a cell, which we'll talk about in our tissue lecture. If you were to take a look, the microfilaments allow that structure to maintain that rigidity on those villi. You're going to see intermediate filaments, and intermediate filaments basically hold these organelles in place. They don't just swish around wherever they want to. They're kind of held in place with these intermediate filaments. Microtubules allow for movement of the cell. You'll see microtubules around there, these big structures that are going to be involved in the cell division, which we'll talk a little bit in a later slide. Now, looking at these organelles for movement, we do have some of the other we have intracellular and extracellular organelles that are specifically geared for movement within the cell or the outside of the cell. You'll see centrosomes. Centrosomes are built inside the cell to allow for an arrangement, and you'll be able to see this later on, an arrangement to allow mitosis, the splitting of the cell, to take place. Looking at extracellular, we have cilia and flagella, and these are things on the outside of the cell. Cilia are things on top of the cell that allow for ore strokes. They're basically allow movement. When we get into our respiratory lecture, we're going to be able to see the epithelial cells that are exposed to the air. And we have cilia that allow movement. And what they do is they literally just move like this. And any stuff that doesn't belong gets captured by the cilia, and it gets it to move away from the lungs. Flagella, on the other hand, allow cells to move. And sperm have that exact thing. It's a flagella that allows these whip-like motions to have it move through a solution. We don't ever really talk about flagella, so don't worry about it. And this 9+3 arrangement, don't worry about that either. That talks about the inherent structure of these microfilaments that clearly not concerned about. But intracellular centrosomes, extracellular cilia, which we'll be able to see in our respiratory lecture, typically found on the top, the apical side, the top of the cells, to allow movement of stuff exposed in that cavity. Now, looking at the organelles that are involved in protein synthesis. The purpose of protein synthesis is what a cell is supposed to do. It's supposed to manufacture proteins. Whether those proteins are geared for the cell itself, geared for the cell membrane, or geared for the outside of the cell, for the production of a hormone, the production of a neurotransmitter, stuff that a cell needs to make. So what we're going to be talking about in this process of protein synthesis is this entire highway where we produce a protein and get it to its intended target. And the first thing that we start off with is the DNA itself, and we'll get into the DNA in a little bit, which contains the blueprints for it. The ribosomes are organelles that are going to start the process of protein synthesis. And our lab today will go over that specifically, and we'll talk about this portion at the end of today's lecture. We'll see the formation of a protein. So we start pairing a bunch of amino acids together, stringing them along, creating, and this is my question for you guys, which would this be? Would this be the primary, secondary, tertiary, or quaternary structure? The string of amino acids.
Primary?
Primary. So this is where we undergo the creation of the primary sequence. If you were to take a look at the ribosomes, they're comprised of 40% protein, right? So they're made from the stuff that they were produced from. And then the other 60% are RNA. This is a ribonucleic acid, our single-stranded nucleic acid string. So they start the process of creating the protein and primary sequence. Now, the endoplasmic reticulum is the next stop. And don't worry about these terms as sister name. The endoplasmic reticulum is a series of membrane-bound flattened sacs. And what do I mean by that? It's basically another highway that's the next stop in the protein synthesis chain that is geared for the synthesis of the proteins, the storage of the proteins, or the transport. So these three main portions. So you'll notice about the endoplasmic reticulum that this one specifically is studded by ribosomes. So it's not uncommon for you guys to find ribosomes that are embedded on top of the endoplasmic reticulum. It serves as a one-stop shop for protein synthesis to take place. So rough endoplasmic reticulum, or rough ER, are covered in ribosomes. These produce proteins. Smooth endoplasmic reticulum are the ones that don't have ribosomes on them. And those are specifically for producing any lipids of any kind. So if you were to see rough endoplasmic reticulum, you know they have ribosomes on there and they're geared for production of proteins. So what's taking place in the endoplasmic reticulum? If the ribosomes are creating the primary sequence, secondary, the folding, the protein folding, is also taking place in the endoplasmic reticulum. So not only are we seeing the secondary, we start seeing the tertiary. We see the disulfide bridges. All that folding that we talked about on Thursday takes place. And then maybe getting quaternary structures, getting multiple subunits to come together. So here you're going through that bigger stage of a set protein assembly taking place in the endoplasmic reticulum. Now, the third stop will be the Golgi complex. This is next in place. So when we had the ribosome, we had the endoplasmic reticulum. The Golgi complex is where we go for the packaging of the protein. And this packaging allows it to kind of accommodate for its destination. So in most cases, this is going to need to make it up to the cell membrane or through the cell membrane. And in those cases, it's going to have to employ vesicles. So this is where if the ribosome is making the protein, endoplasmic reticulum is assembling it and putting it together, the Golgi apparatus is, for all intents and purposes, just wrapping that package together. So it's that final wrapping to make its way to the cell membrane. The Golgi apparatus not only will wrap it together, but it'll also modify that protein by adding carbohydrates to create things like glycoproteins. Take a protein and then attach those carbohydrate signals up on top. And if I can go back to our earliest slide, I know I'm going back like 20 slides. We can see proteins here embedded with these carbohydrate chains. So this is what the Golgi apparatus does, is by adding these additional features to the protein. And to kind of sum that up, here you can see the ribosomes embedded on top of that rough endoplasmic reticulum. You'll see the ribosomes are creating the proteins. You'll see that eventually it'll make its way over to, this is the endoplasmic reticulum. It's going to get packaged within. And then it's going to make its way over to the Golgi apparatus where it can get packaged. Here you see that protein. You see the protein getting modified. And that protein can make one of three pathways. A secretory vesicle where it can get secreted out of the cell, where that protein can become part of the cell membrane, or this last one, the transport vesicle, to the lysosome, where it can get destroyed. So with that, let's take a look at that example. So if I were describing this path right here, the secretory vesicle, this is an example of exocytosis. This is where the protein that's made and packaged by the Golgi apparatus makes its way out of the cell. Exocytosis, the exit of that protein. And this is a video showing you how that's done. You'll see some docking proteins that kind of creates it and opens up that vesicle and allows it to fuse with the plasma membrane. And you see that dumping of that contents out into the extracellular fluid. Let me play that again. I know it's only 12 seconds. Just in case you didn't get that. You'll see where this comes into place. And we'll talk about this in our neurotransmitter lecture, where these docking proteins come in. You see the opening up of that vesicle, and you get all this stuff dumped out. So the example of exocytosis that we'll get into are neurons that are going to be secreting neurotransmitters to make its way to the next cell. And that's exactly what's taking place. This whole process.
So that process was from ICF to ECF, right?
Yes, ICF to ECF, yes. So the production of the neurotransmitters, which are going to be, not surprisingly, proteins or amino acids, are going to go through the process of creation, production, to packaging, to make its way out of the cell itself. So that's a process of exocytosis. What questions do we have about that? The other organelles that are involved, we're going to see our cleanup crew. These guys are not involved in the protein synthesis, but they do exactly that. They clean up the cell. We're going to see lysosomes. Lysosomes are structures that contain digestive enzymes. These are probably one of my favorite ones, because what they do is they destroy things. That example that I pointed out earlier, way earlier here, lysosomes are going to have these digestive enzymes that come in and destroy foreign bodies. They don't recognize it and they don't have those cell signals. They come in and they destroy it. So either through endocytosis to drink whatever's been taken in, or phagocytosis to take in a larger body. It'll come in and destroy it with those enzymes. What's interesting about those is a disease called Tay-Sachs disease. And again, when they look at Tay-Sachs disease, this disease lacks these lysosome enzymes. So you get the buildup of stuff that takes place in nerve cells. We have nerve cells. And over time, there's components within the cells that start to degrade. Maybe you get ribosomes, maybe you get endoplasmic reticulum or Golgi apparatuses that no longer work. And eventually, if they don't work anymore, well, let's destroy them, come into the cell, and destroy it altogether. So if you have an endoplasmic reticulum, destroy it. Take that protein that makes up the endoplasmic reticulum, chop it up into a bunch of pieces, into amino acids, and reuse it for the cell. Unfortunately, what happens with Tay-Sachs disease is the mutations that are involved in the creation of these digestive enzymes. Remember, these are enzymes that are encoded by proteins, or from proteins. Proteins that have a specific function to destroy. You remove that mechanism to destroy, and now you have all these old cell pieces that are floating around. And that eventually causes dysfunction of that cell. You've increased the amount of solute, stuff that's dissolved in the cell. And eventually, water will start making its way in. And that'll start to render those nerve cells as dysfunctional. So simple things, any mutations that come in. And again, your lab today will go over what happens in the cases of mutations that can cause dysfunction. And one disruption of one major organelle can cause degradation of the entire cell. Now, peroxisomes, the second on here, are oxidizers. So they oxidize and remove, and we'll again talk about this on Thursday, they remove hydrogen from a molecule. In this case, this is their way of removing things that shouldn't be in our systems. Give you a good example is the metabolization of alcohol. We have peroxisomes that come in and take alcohol and remove hydrogens on there to turn it back into water. Proteasomes are another proteases. They're basically a structure that contain enzymes that break down proteins. So when proteins are no longer functional, we degrade them and break them apart. The next one I want to go over are our power cells. Again, the thing that we're going to be spending a good amount of time on this Thursday. We'll be talking specifically about the mitochondria. And what's interesting about the mitochondria, well, let me first talk about what it does. It's a powerhouse. It generates ATP. It's going to be producing a lot of ATP through the process of cellular respiration. And what it ends up doing is it takes glucose and takes the byproducts of glucose and in the presence of oxygen helps this production of ATP. And again, we'll go into more detail on Thursday. If you were to take a look at the mitochondrial structure, you'll see that it actually has a very different structure. It has an outer membrane and an inner membrane. So it has two membranes as opposed to the one cell membrane that cells have. Within the inner mitochondrial membrane, you'll see these folds called cristae, and they increase the surface area of the membrane itself. If you were to look at the inside of the inner mitochondrial membrane, you're going to have what are called matrix. It's a fluid-filled structure, and there's going to be an importance when we learn about the inner layer as well as the fluid on Thursday. Now, what's interesting about mitochondria is that it's theorized to have had origins from a prokaryotic bacteria that was swallowed up by eukaryotic bacteria. They came in, and that symbiotic relationship allowed this cell that used to independently operate to produce ATP and get swallowed up by a cell to protect the mitochondria, at the same time receive ATP from the mitochondria itself. So mitochondria existed within the cell, and they find that the DNA that's contained, the mitochondria actually has DNA of its own. There's mitochondrial DNA. It's actually from your mother. So if I were to take a look into your cell and grab that DNA from inside, it's actually maternal DNA, as opposed to DNA which is supposed to be a mix of both mother and father's DNA. Now the nucleus is what's going to contain the DNA, the blueprints of the entire factory. And let's look at how that takes place. You'll see the nuclear envelope separates the DNA from the cytoplasm. So you're going to get this, you're going to get a cell, and on the inside of the cell, you're going to get the nuclear membrane. And on that nuclear membrane, you're going to see the pores that are present on top of that nuclear membrane. And those pores allow stuff to move in and out. We'll talk about what moves in and out in just a second. On the inside, you're going to find your DNA. And our DNA is going to be double-stranded. If you're to take a look at the DNA inside, what most of you guys have seen what DNA looks like, you would have seen DNA looking like this, right? But this is not DNA in its natural form. In its natural form, it's actually formed as chromatin, meaning DNA, if you were to take a look at the DNA itself, it would just be this jumbled mess. And I call it a mess. Actually, it's not really a mess. There's this kind of inherent structure. This is called chromatin. And when a cell starts to prepare for cellular division, meaning that cell starts to separate, it has to organize that chromatin into chromosomes. So the organized DNA that you see, that you've seen before, are chromosomes. And this is the process of which you get cells to divide and to organize and divide accordingly. So this you don't really have to wholly understand, but here you have your DNA, and if you take that jumbled mess of DNA and you start to prepare for cellular division, what happens with that DNA is it starts to wrap itself around these histones. These are these proteins that are present, and it allows that DNA to wrap around in this organized fashion. And eventually, once they start to organize together, they can get put together into what's called a chromosome, of which I'm a center. You don't need to know this term. The centromere is the anchoring point for two chromatids, which are going to be, one side is going to be the mother's DNA, and the other side is going to be the father's DNA. Actually, that's drawn this way. So that is the organization. So chromatin, then organized into chromosomes. Now, if you're to take a look at what we do with that DNA, once with DNA in the process of developing that protein, the main central dogma of protein synthesis is that we take DNA, we're going to be able to convert a DNA strand, I don't want to say convert, but copy a DNA strand into an RNA to eventually create into protein. So DNA, think about the analogy that I'm going to use, is that DNA is going to house the entire contents. These are going to be the entire blueprints for how a cell is formed. If that entire library, what we're trying to produce is a protein, a single protein out of it, what we need are blueprints just for that protein. So what's going to take place is we're going to have this entire, and if I can just draw our DNA simply drawn as such. What we're looking for is a particular gene. And a gene is a specific marker point from one side of the DNA to further downstream for another point. This represents a gene. Now, if the DNA right here is the entire library, RNA is like taking a Xerox copy or a photocopy of just one section. So the gene represents the area that we want to target, and what we're going to do is we're going to create a single strand from that gene. And this is going to become our RNA. That RNA is going to undergo some modification so that it can create the blueprints for that protein. So that process of when we go from DNA to RNA is transcription, and you guys will be doing this in the lab. You're going to transcribe that DNA into RNA, and all of that's going to take place in the nucleus. And then when we go from the RNA to the protein, this is going to take place in the cytoplasm, outside of the nucleus. And what we're doing from here, from RNA, we have our nucleic acids, and we're going to translate that into amino acids that are going to form a protein.
Wait, can you repeat that
Yes,
again?
of course.
It was to amino acids. What was the word before that?
Translation. We translate the nucleic acids
Nucleic
inherent
acid.
in the RNA structure and translate that into proteins. I'll get into that right now. That's just really the central idea of what's happening. What we're going to be taking a look at next is the process of transcription. This is transcription, and this is translation. And I know there's a lot of text on there. So let me describe what's taking place. We're going to take a look at that section of DNA. Like I said, that long library. And we're identifying one gene. And a gene is that point of which the protein, we want to create the protein from. As you saw in that last slide, one gene equals one protein. So to give you an example, the genetics behind your eye color is looking for the pigment, which is a protein in the eye. So the gene that encodes for that color is embedded somewhere in your DNA. And I don't really know where, I'm not a geneticist, but at some point in the human genome, we've identified where that point is. So if we want to create that pigment, we've got to identify it in that DNA. So we take that DNA and we're going to create an RNA template from that. Basically what we're going to be doing is splitting that DNA so that we can expose, and if I could just zoom in real quick, we're going to expose the bases, the nitrogenous bases, on one side of the strand. And if we were to take a look at the other side of DNA, the other strand, this is going to contain the other sequence, the complementary sequence. A binds to T, T binds to A, G binds to C, and C binds to G. So this is the complementary sequence. We're going to take this portion right here, and we're going to transcribe this into RNA. And so what is involved is an RNA polymerase, an enzyme that's going to create an RNA and string a bunch of nucleic acids into a polymerized form of RNA. And it's going to recognize the very first portion of that sequence. And again, your lab will go into this. It'll look for a promoter. A promoter is an area that's going to be the starting sequence. And then it's going to build an RNA from it. So it's going to create this RNA, which I'll draw in a different color. We'll draw it in green. It's going to create the sequence from this. Simply put, it's going to turn this into the exact complementary sequence from earlier. Except where you see A is supposed to bind to T, in RNA there is no thymine. It actually binds to uracil. So this was the fifth, uracil was the fifth nucleic nucleotide in RNA. So here we're going to create U, A, C, G. So this becomes this template. So this becomes RNA. And it actually carries out, it could be extremely long, but now you get this really long complementary sequence that's single-stranded. And eventually, it'll cut off at a certain point. So now that you've got this long sequence of RNA that's single-stranded that starts with U, A, C, G, U, A, G, C, I'm just going to string a bunch of random letters, U, C, U, A, G. You get this really long sequence that's copied from the DNA. And now what we're going to do is going to undergo a process where we're going to be able to cut out certain things that are not relevant. So we're going to find pieces on here that might not be what we're looking for. And so we have small nuclear ribonucleic proteins, also otherwise known as SNRPs, RNPs, and they come in and excise areas that we don't need. So they might find that this area might not be necessary, and they'll kind of bind them together. So this area might not be necessary. They call these introns. These pieces are called exons. And then what these SNRPs are going to do is they're going to cut out the introns and put these together. So the final product is going to be UACG, UCUAG. And this is the template that we're going to create proteins from. If you might be asking, why are we cutting out pieces? Well, SNRPs are geared to cut out pieces. They are specifically attuned for certain sequences to cut things out. Because this gene right here that we were able to copy from that section over there that we created the RNA from, this could be encoded for pigments.
How does it know what to cut out though?
These SNRPs are specifically geared for that. They look for a specific sequence, they see it, they cut it, and they make the cut.
And then what happens to it?
Shorter.
Just destroy it, we just get rid of it. We're only cutting it from the template. We're not cutting it from the DNA. We never want to mess with the DNA itself. We just want to cut it off the background. Now, why would we want to cut this out? This might be the code for pigmentation, a specific pigment. There are other SNRPs. We could use this RNA for a different protein. So a different SNRP can come in and say, don't cut that piece out, cut from here to here, cut that piece out. And now we have something like another protein. So in other words, what I'm trying to get at is the sequence that comes out from that RNA can encode for multiple different proteins, depending on where those SNRPs cut. This SNRP that cuts for pigmentation will cut over here and here. This SNRP will cut and create something of pigmentation for hair. So if you were to take a look at the DNA, we can take, if we were to take a look at the 30,000 genes, you guys don't need to know this, the 30,000 genes that we have, we've got a lot of DNA. We take 30,000 genes, locations on the DNA. We can create something on the lines of like 1 million proteins from that, just based on where those SNRPs cut. So your labs don't go really much into that. I'm just getting into the detail to kind of give you an idea of how complex our DNA is, but how we're also able to conserve all that DNA language that comes out of it. We can take an RNA, cut it out into pieces. Now we get what's called an mRNA. An mRNA is the final template. It's the building block which we're going to turn that mRNA into a protein. So while we've been able to transcribe DNA into RNA language, now we're translating that RNA into the protein. And what you're going to notice is in that sequence, UAG, UCU, AG, UCAGU. This is our mRNA. What we do with that mRNA is we're going to look at triplets. And we take the next three pieces of each of these, and let me just finish this off, UAC. We're going to create triplets of nucleic acid, nucleotides. And these nucleotide sequences, these triplets are called codons. And each of these will encode for an amino acid. And if you just take a look at your labs, what I'd like you guys to do is to take out your labs that I gave back today, and take a look at the last page. And you'll see the mRNA codon amino acid chart. How many amino acids do we have?
20.
We have 20 amino acids. And if you look at the different combinations that we have of first base, second base, third base. And what do I mean by bases? First, second, third. With the different combinations that we have 64 different combinations. You'll see that a codon, a triplet sequence of RNAs, correlates to one specific amino acid. So if I look at UAG, and you look at that chart, you look at the first base, UCU. U, second base C, and then the third base, UCU, you would get serine, right? So that would be the amino acid that gets formed on top, the amino acid being serine. And if you were to look at the other one, that would be serine. If you were to look at AGU, A, the second base, G, and serine again. If I look at CAG, glutamine, great, thank you, something different, finally, glutamine. And if I look at UAC, tyrosine. Okay. That's exactly what you guys are going to be doing in your lab, is you're translating these codons into the amino acids. So now what we have is serine, 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 glutamine, and tyrosine. That is our primary sequence.
How come for serine there's 2 and there's 4? Is that just different, like you see, or am I looking at it wrong?
So your question is, why does it seem like serine has a bunch of triplet codons?
Yeah.
So this is something that will be answered when you do your labs. I'll answer it for you now. But what you're going to notice is that there's some redundancy. You'll notice that UCU, UCC, UCA, and UCG all encode for serine. And it looks like serine, you also saw AGU and AGC. So any of those triplet codons will still create serine. So there is a lot of redundancy in our DNA, in the nucleotide sequences, to produce that amino acid. If I have, again, 4 times 16 times 4, 64 different sequences. 64 different sequences available. So if I have 3 positions, I have 4 available positions for 4 different nucleotides for the first base, times 4 available for the second, times 4 available for the third. That comes out to 64 available combinations that you can make from all of those to encode for 20 amino acids.
The amino acids are the ones in brackets, right?
The ones
They're
in parentheses. Phenylalanine, serine, tyrosine, cysteine. Those are the ones on there. You see that? So the redundancy is present so that if there's ever a mutation, what kind of mutation am I talking about? Some, I don't know, sun energy. Or maybe a mutation that occurs during, there's skip mutations, there's phase mutations, all different types of mutations that could come in and destroy something in the DNA and completely remove or add in or completely cause different types of mutations that disrupt the sequence. And again, when you go through the labs, you're going to see different types of mutations. You're going to see a frame shift mutation. You're going to see a point mutation. That could potentially change the sequences after. So if this becomes, instead of AGU, turns into something completely different, and it becomes thiamine, or creates methionine, that disrupts the primary sequence of this and can completely change the configuration of the protein. So therefore, there are employed redundancy involved to allow mutations that could result in no changes in the protein at all. So 64 different combinations for 20 different amino acids. And we will see that. What questions do we have about that? So now we talked about translation. Let's sum it all up for how this process is built together. And that I can show you on the next slide. So what happens on that slide is that the mRNA needs to bind, the mRNA binds to the small ribosomal subunit. So here's our mRNA sequence. It comes in and binds to the small ribosomal subunit. And we're looking specifically for an initiator tRNA. But in this process, what's going to happen is we have an initiator tRNA. It's called a transfer RNA. And what a transfer RNA does is it carries an amino acid with it, and it has an anticodon. And that anticodon is meant to bind to the complementary sequence of RNA. So if I find a sequence on there called AUG, that tRNA is going to bind together. You're going to get the small ribosomal subunit that's going to bind to the mRNA. This large ribosomal subunit is going to bind together. And there's three sections. There's an E, there's a P, and behind this is a P, and an A site. So here you're going to see the initiator tRNA, and it needs to find the sequence of AUG. It's the starting sequence on an RNA. That initial tRNA is going to have that anticodon with the complementary sequence of AUG, which is UAC. So this tRNA comes in and binds to AUG, the initiator sequence, and it carries methionine. Then this P site, once this connects, this is going to get ejected over to the E site for the exit of it. The next subunit, if I were to take a look and just kind of add another sequence on there, UCU, it's going to have another tRNA that's going to come in and bind, and that tRNA is going to have an anticodon of AGA. So it'll come in and recognize the sequence, bind together, and that tRNA is going to have serine on top. So what that will do, it'll string serine and methionine together. So while I'm describing that, I might be losing some of you guys, so I'm just going to show you this animation that comes in. It literally will just do this. It'll string them together in this fashion as it makes its way through that RNA. You guys see how that works? So once we start having this, we have methionine, we have our next one, we have this whole string of amino acids that build together. This is what's going to create this primary sequence. And this whole string of amino acids becomes exactly that. This is the foundation of our protein. So that's what you guys are going to be doing in the lab. In that lab, you're going to be able to, you're given a DNA strand. You're going to convert that DNA into RNA. That RNA is going to get translated into a protein. And you'll learn about what mutations can come in place that will affect the primary sequence. Because then you take that sequence, it becomes a very long sequence, and then it's going to get folded up, it's going to get put together in the endoplasmic reticulum, it's going to get folded up in the and packaged by the Golgi apparatus, and eventually it makes its way over to the cell membrane or out into the extracellular fluid. That way, that pretty much sums up the entire process for the production of proteins. This next bit, we're going to look at cell division. I want to just kind of touch on key points so that you'll understand what we're doing here. I'm not going to go into too much detail about this, honestly, because we're not really going to go over mitosis and meiosis. Again, this is mostly cellular physiology that, in my opinion, we can go into a lot of detail of, but I don't want to derail us from the main idea of what's taking place. So I'll give you the sort of executive summary of it. Is that okay with you guys? Yes. Cool. All right, so cell division. Why are we dividing a cell a bunch of times? Well, the cell can divide itself when it needs to. Let's say we're undergoing heavy growth. Let's say you guys are going through puberty. You guys are going to be growing, you're going to get a lot of bone growth, you're going to also get a lot of skin cell growth, and therefore cells need to start dividing. They don't just continually divide, there's certain markers that it has to reach that allows it to start dividing. So in this process, what it's doing, it's going to go through the process of cellular division. And there's two things that need to take place when you're dividing. Think of that factory example that I was talking about earlier. A cell is a factory. And if you're employed, you're there to manage the division of that factory into two functional parts. You've got two things to take care of. You have the DNA, which is going to contain your blueprints. Okay? And the other portion, you need to make sure that you have enough machinery on both sides. And what do I mean by machinery? I'm talking specifically about the organelles. So if you were to think about the division of it all, most of the time that a cell spends in that cellular division is all through the preparation of that division. It needs to duplicate the amount of DNA. It needs to create enough organelles and make sure that we have enough proteins. It needs to make sure that it has all the pieces that it needs first before you divide. It's like moving. When you go through the process of moving someplace, I mean, you've got your place that you've selected, but the whole process of moving involves categorizing everything, putting them into boxes, make sure you have enough that you can move over to the other place. That might be the best example. This is one of those moments where I think, okay, maybe that analogy isn't really working that well. But nonetheless, there's a lot of time in the preparation before you actually go through the division or the process of moving. So in this case, looking at this whole process, this entire clock right here is that process where we go from one cell to two. And the entire process of the cell splitting is the mitotic phase. And that is the process of mitosis is the process where we actually undergo the division of the cell. But prior to all that, where we spend most of the time, we actually start from this point right here. This is our three different phases called interphase that takes place prior to the cell dividing. So interphase is divided into three parts, G1, S, and G2. And you would see that G1 phase is where the cell becomes increasingly metabolically active. It's producing a bunch of organelles. Again, if it's producing proteins to become those organelles, it's taking that DNA and creating mRNA and turning them into proteins. It's creating more centrosomes. It's creating more ribosomes, more endoplasmic reticulum. So it's continually churning out and doubling everything in itself. The next portion is S phase. And this is where it's going to take that DNA and duplicate that DNA, because we need one copy for one cell, we need another copy for the second cell. G2 phase is where cell growth continues even further. We're now creating more proteins, more enzymes, and even more so organizing the cell. So these three phases, if you were to total up the amount of time during that, that's about 20 hours that's required for the actual cell, for the preparation of cell division. The last phase is a mitotic phase, and we'll talk about that after we look at DNA replication. DNA replication, to quickly sum this up, we get a DNA polymerase that comes in. It literally will splice the double strand, find the complementary sequence, and create a new DNA from it. It's unzippering that DNA, coupling that new strand together, and creating that second sequence for cell number 1. This one's for cell number 2. The organization of it, we're taking the chromatin, which is this, and I like to use the example of a bowl of spaghetti noodles. That's the DNA that's kind of in this bowl of noodles there. And you're going to get the chromatin that's going to get organized into chromosomes. And when you look at the chromosomes, you're going to see that they get organized together. You have the centromere, which divides the left arm with the right arm. This gets organized into what you've seen as the 23 pairs of chromosomes. And this is what we call our karyotype. You don't need to know that term, but this is what you've seen before. This is the human genome in DNA that is organized prior to cellular division, of which the first 22 are autosomes. Again, you don't need to know this. Autosomes, which are going to characterize everything else besides the sex of the cell. So first 22 are autosomes. These are your sex chromosomes, of which the sex chromosomes determine the sex of the baby. Other things that I want to point out, telomeres at the very end. Telomeres, they're redundant sequences of DNA, but you'll find that if any of you ever have an interest in gerontology or aging, you'll find that telomeres are important. These are redundant sequences that are all geared to protect the chromosomes. So this contains the DNA, the blueprints. These right here are the buffers, because we've got enzymes that come in and can sometimes cause mutations or chop off pieces, and telomeres serve to protect those. And if you ever look at aging, and what happens with us as we start to age, and all of us here are aging, over time, these telomeres are shortening. these are breaking down. So you'll start to see that, especially people that become older, well, obviously everybody becomes older, but eventually as people become older, mutations can start to happen on the DNA itself and start to cause things such as cancers and things that manifest much later in age. The process of mitosis, again, looking back at what's taking place. Mitosis, at the beginning of mitosis, we've already duplicated everything. The organelles, the enzymes, the DNA. Now we're going to start to prepare for the division. And we've knocked off a couple terms already. We've talked about chromatin, chromosomes. What we're going to do in mitosis is separate everything out. So this is the last phase of interphase, where we take the chromatin and we convert it into the chromosomes that you saw earlier. Now prophase is the very beginning. And you're going to see that we have 4 different phases of mitosis. Prophase, metaphase, anaphase, and telophase. Otherwise known for my third mnemonic now, PMAT. And in this process, what you're going to see is we see prophase taking place. You see our chromosomes starting to somewhat organize together. In prophase, we start to see the breaking down of the nuclear membrane. We start to see centrosomes creating the north pole and the south pole of the cell. And those centrosomes come in together to create these mitotic spindles that serve as the skeleton to divide the south pole from the north pole. So this creates polarity of the cell. The next phase is actually an intermediate phase called prometaphase, where the nuclear envelope is almost broken down. You get the microtubules that are coming out of the centrosomes, the north pole and the south pole here, and they'll start to extend those microfilaments onto the chromosomes themselves. You don't worry about the terms kinetochore or non-kinetochore microtubules. Basically, all it's doing is it's leading itself to the next phase called the metaphase. And what it's done, it's taken all that DNA that you've duplicated and arranged them onto a metaphysial plate. That term. It's a line right in the middle. If you see, this is the north pole, this is the south pole, this is kind of that equator of the cell where the DNA is lined up. In anaphase, anaphase serves to pull those sister chromatids apart. You're basically pulling those DNA that's duplicated and now pulling them onto both poles, on each side of the cell. So after anaphase, once you've actually separated those DNA apart, now you're going to undergo telophase. And telophase starts the reversal of what's taking place. Telophase arranges the DNA, and cytokinesis is the process where you actually split the cell. You have what's called the cleavage furrow, and there is microfilaments that are involved in cleaving the north pole from the south pole. The way it works, it's like a drawstring that wraps around, and it comes in and draws that drawstring tight and eventually squeezes to the point that they separate. And now you have one cell with equal amount of DNA, and you have another cell that's equal with amount of DNA. This is the process where we divide the DNA apart and create two cells. So now in the end product of mitosis is we get two identical cells with the same amount of DNA. You turn one cell into two. Meiosis. Meiosis is important in reproduction. And since we're not talking about reproduction at all, it's not even really all that important for now. All we're doing is, in meiosis, is we're creating a cell, we have a cell that has the blueprints for a DNA, and we're creating them into what we can call half cells. So while the DNA is duplicated, we have 2N, we have duplicated DNA on one cell, we're going to eventually divide twice to create half cells, cells with half the DNA. And these are called gametes. What are gametes? sperm and eggs. These are the gametes that we're turning normal cells back into gametes to be used in reproduction. So in this process, we divide twice. There's some key terms on here: haploids, crossover, chiasmata, and independent assortment. What takes place in meiosis 1, we see the DNA get jumbled up together. We have chromatin, turn that to chromosomes. We get chromatids that come together. And the key difference in this case in prophase is to metaphase is that we have 2 pairs of DNA. And here we can call, just for the sake of simplicity, the blue ones will be the paternal DNA. The red one will be maternal DNA. We get that duplicated. I'm sorry, not duplicated. We do duplicate them, but we arrange them along that metaphase plate where we have pairs that come together. Independent assortment is where, the crossover is where you actually get paternal DNA that mixes with maternal DNA, and you get them to mix together. Independent assortment is the arrangement of that DNA. So this represents chromosome 1, this is chromosome 2. You actually go all the way up to chromosome pair 23. And that all gets arranged together, and independent assortment is either flipped this way or it flips the other way. So you take all 23 and you kind of arrange them in random order. And we go through metaphase, we go through anaphase where you split, and now you've got DNA on one side, you've got DNA on another. The next thing we do, we take that same that last branch right here, we take that and we split it again. So now we've got our DNA, we split in one direction or we split in the other direction. So in that process, you can see what happens with this DNA. It undergoes prophase, metaphase, again, independent assortment, whether it flips this way or flips the other way. We go through metaphase, we go through anaphase, taking them apart, and then telophase, and we split them again. And what we end up with are haploids. Half the DNA of what we started. So that process, where we have that half DNA, that mixes with the other half DNA of another person, a willing partner, those come together, and you mix the DNA together. And that process you might be familiar with, and that's how you basically combine the DNA together. So that becomes the new maternal DNA, the new paternal DNA, and that allows us to add genetic variety to the DNA of an offspring. So these points right here, crossover and independent assortment, again, not something I'm really going to talk about too much after this. Could be part of the quiz, but after this, don't even worry about it. Okay? Hence why this is the cutoff point. If you want to see how these take place, you can see the crossover and meiosis. This goes into a little bit more detail about how crossover takes place. You guys have this in your slides, right? Yeah, okay. And then independent assortment is that process where you can get the arrangement, the 2 different possibilities of arrangement of DNA that can get rearranged in another fashion. So this is the summary of the both of the two, where mitosis, you get 1 copy turned into 2 copies. Meiosis is the splitting. You take DNA and you combine that into, you turn them into half DNA. So far we've looked at, in this process, we've been able to look at how we're able to eliminate byproducts. We talked about the elimination of byproducts through lysosomes. We've talked about the synthesizing proteins through the process of transcription and translation. And we've talked about the exchange of materials between the environments, ways to get things in and out of the cell through carriers, through channels, and so forth. We're going to go into more information about material transport later on. We've also talked about reproduction. So these pieces right here, we've talked about a good chunk of general cell functions. We'll start to get into much more detail about each of these, especially when we get into our specific organ systems when we get into them. We'll see the general cell functions and what they do. We'll talk about step 1 and step 2 on Thursday's lecture, and then response to change in the surrounding environment. This is the whole concept of homeostasis. We'll be able to visit how these take place in the different organ systems, what happens when changes take place, and what we do to accommodate with. So with that, that wraps up today's lecture.